Hello and welcome back to Academy. So till now we were discussing about the different stages of cell cycle and the regulation of cell cycle. We have also talked about what are the regulations, key events which helps in the entering into the mitosis. In this lecture today we are going to talk about what are the different stages of the mitosis. So this already we are learning from our 11th and 12th standard. We have also studied in our graduation level and post graduation level. So just let us just revise it. So cell cycle is highly ordered and tightly regulated. So this we already know because this is what we have been learning in throughout the cell cycle series. So we know that in case of prokaryote is the binary fission, or it is also called amitosis. So we know that what is binary fission? It means that from one bacteria when it divides you get two daughter cells. Okay, identical daughter cells we get. Now, in case of eukaryotes, there are two main stages of the cell cycle. So this we have already said, that is the interphase and the mitosis. So where interphase has been divided into G1 phase, S phase, G2 phase, and then the other is your M phase. So altogether the cell cycle in eukaryotes takes around 24 hours approximately. Now, in today's lecture, we are going to talk about the different stages of the mitosis as already mentioned. So what are the different stages of mitosis? The first one is prophase, metaphase, anaphase, and telophase. Now, further after the telophase, that is towards the end of the telophase or the late telophase, it is followed by the cytokinesis. Now, what is cytokinesis? It is the production of two daughter cells which directly goes into the G1 phase of the cell cycle. Okay, so now we are going to talk about the first, and that is the interphase. So overall, we have studied about this. Just let us just summarize it. So interphase is the period between two mitotic stages. So before I proceed, let me just tell you. So I was not able to type entire thing. So I got the directly notes, and these notes have been taken from the Let's Talk Academy. So the thing is that notes are given very nicely and all, and more of the explanation will be given by me. Okay, so let's just continue. So interphase is a period between two mitotic stages. The chromosomes are decondensed and dispersed throughout the nucleus, and it is in the form of chromatin. So we know that, like earlier lecture, we talked about that whenever the cell is entering into the M phase, what happens? The nuclear envelope degrades, your chromatin condensation starts, etc., etc., right? So the first stage of the interphase is G1 or growth stage 1, which determines the fate of a cell. That is, either the cell can undergo differentiation and enter in G0 phase. So we know that what is G0 phase? That is the resting phase, or it is also called the quiescent state. Or it can proceed to the G1 into the replicative S phase and commits to complete the entire cell cycle. Okay, so now duplication of genome is restricted to S phase. So S phase means, this means what? It means that they are talking about the DNA replication. Okay, here S phase, they are talking about the DNA replication. Now the second gap phase, that is G2, is the last period preceding the initiation of the mitotic prophase. So we know that we have got the G1, S, G2, and then M. Okay, so this is how our cell cycle looks like. So the duration of these cell cycle phases varies with different kinds of cells. This is also we have mentioned. Now, during the interphase, the cell is prepared either for the mitosis or meiosis. So about meiosis also, so we know that mitosis and meiosis is also called the reductional division. Somatic cells undergo mitosis to reproduce themselves through cell division, while germ cells undergo meiosis to produce haploid gametes for the purpose of sexual reproduction. So this we will talk in about the next lecture. So here you can see that, so the cell is in the interphase and the chromosomes are getting replicated. Sorry, no, the chromatins are getting replicated, the DNA is getting replicated. By that time, the centrosomes are also getting duplicated, so that it will move towards the opposite poles. Now, the first phase of the mitosis, that is the prophase. So it is the early prophase, chromatin fiber gets condensed to form the chromosome, right? So in the earlier lecture, we have talked about how it gets condensed. So what are the proteins which is responsible for the condensation to chromosome? Can you tell me? So we know that before condensation, the proteins like cohesins gets phosphorylated, because of which they get removed, right? So remember, we had our chromosomes like this, and to that your cohesin proteins were there, which were keeping or interlinking the sister chromatids. Now later, when it enters the M phase, the proteins called condensins gets activated by the activity of CDK1 and cyclin B. If you remember, in the previous lecture we have discussed about this in detail. Okay, then the nuclear membrane degradation takes place. So here also, remember, the lamin proteins like A, B, and C, even they get phosphorylated by the activity of CDK1 and the Aurora B kinase. So which will result in the degradation of the nuclear membrane, because once the lamin proteins gets phosphorylated, it is not going to form the dimers and hence the membrane. The lamin protein gets free from the inner nuclear lamina to form protein scaffold onto which chromatin fiber loop out to form the chromosome. Now, vesicles gets generated due to nuclear membrane degradation remain present in the cytoplasm. So, what does it mean? So, when we say that the nuclear membrane that is getting fragmented, these membranes actually results into the formation of vesicles like this. Okay, so these vesicles you can see into the present in the cytoplasm like that. And later when the cells is getting divided, that is two daughter cells are going to form, then these same vesicles will go and attach to the chromosome and will form the again the nuclear membrane back. Okay, now nucleolus disappearance also takes place in the prophase. Please remember this also, such question comes for the 1 mark question. So this is also important that nucleolus disappearance takes place in the prophase of the mitosis. Now microtubule formation restores in the prophase. Now why this is required? Because these are going to form your spindle fibers, which will go and bind to your centromere of the chromosomes. Okay, now centrosome gets split into form to form the two centriole, which moves towards the opposite pole, and the astral rays formation from the central gets started, and thus in late prophase spindle apparatus formation gets started. So if you see the figure over here, you can see that the centrosomes are also getting splitted into two centrioles, which is moving in the opposite direction. So one is going to come here, one is going to come here, and your chromosomes are finally condensed, and it is forming the chromosomes, complete chromosomes, the condensed form of the chromatin, which is consisting of two chromatids. Okay, and then they are held together with the help of the centromere at the center, and you can see that the nucleus is also getting disappeared. Now, during the late prophase of the cell cycle, you can see that there is a appearance of the spindle fiber, and these spindle fibers will go and bind to the kinetochore of the chromosomes so that they can hold the chromosomes. So in the late prophase, microtubules begin to search for kinetochore. Kinetochore is a motor protein attached with the centromere chromosome. And these kinetochore provides an attachment site to microtubules as already discussed. Now coming to the metaphase. So what is metaphase? So already like in the when we were in 11th and 12th, we have done the onion peel experiments, right? If you remember, we have done the onion peeling. Onion peeling. So whenever this peel was there taken and it was stained with the help of the saffron dye, so you must have seen that the onion peel cells, like this very nicely brick kind of things, they are layered, all the cells are layered, and you can see that the different cells are in the different stages of the mitosis. So you few cells you can see that they're in the prophase, or you can see that the chromosomes are aligned in the center plate, that is they are in the metaphase, and sometimes you can also see that the chromosomes are being pulled to the different or the opposite directions, that is they are in the anaphase. Okay, so such can be seen with the help of the onion peel experiment, which we have done in our 11th and 12th standard. Okay, so in metaphase, centriole it's reached at the opposite pole and the spindle apparatus gets established. Now at the pole, centriole will give rise to three types of microtubules. So here you need to remember this. So three types of microtubules will be there. Those are called the astral microtubules. Then you have the kinetochore microtubules. Okay, and then you have got the polar microtubules. Okay, it is also mentioned over here also, my kinetochore is there and polar is there. Now First, the astral microtubules, it remains in the cell cortex. So what does it mean? It is not binding to any chromosomes, okay? It is not for the chromosome. So they are present in the cytoplasm of the cell. Second, we have got the central also give rise to kinetochore microtubule, which gets attached to the chromosome at the kinetochore, okay? So these kinetochore microtubules will go and attach over here near to the centromere at which the kinetochores are present. Okay, so what is the kinetochore actually? It's a protein structure which is located at the centromere so that the spindle fibers can come and attach to the kinetochore. So by this mechanism, it is able to capture and arrange them in the spindle apparatus at the metaphase, or the imaginary plate at which the chromosomes are aligned in the single line. Okay, now then you have got sorry, then you have got the polar microtubules. So polar microtubules, these microtubules will be just crossing the each other from the opposite direction. So this all actually maintains the shape and as well as the spindle apparatus. Okay. Now the two centrosomes begin pulling the chromosome towards the opposite end of the chromosome. So this we have discussed in the earlier class that it is because of the microtubule motor proteins. Those are kinesin and dynein. So we know that dynein moves towards the negative and kinesin moves towards the positive. That is towards the periphery. This is towards the center. Now it is the action of these motor proteins that the chromosomes are being pulled towards the centrosomes, or that is or to the opposite end of the chromosome. Okay. Now because of this, what will happen? And they will try to move towards your opposite directions, and thus at the end of the metaphase it is going to the anaphase where the chromosomes are going in the opposite directions. Okay, now the third type of microtubule, as already mentioned, is the polar microtubules. They are formed from both sides, overlap each other, they help in the chromosome movement. So actually it is the polar microtubules onto which the motor proteins like kinesin and dynein are moving, holding the chromosomes into the opposite direction. Okay, so remember in the previous lecture we have talked about the frictional force. It is the same mechanism by which the motor proteins are able to push the chromosomes towards the centrosomes. Okay, so they are walking actually on the polar microtubules, pushing them in the opposite direction and moving towards the centrosome. So this is how the chromosomes are being pulled towards the opposite pole. Now coming to the anaphase. So till now we have discussed about how the chromosomes are being condensed. The nuclear membrane gets fragmented, and the kinetochore microtubules goes and binds to the centromere of the chromosome so that they are aligned in the single plane, and later they are being pulled towards the opposite directions. Okay, so that is the anaphase. So the chromosome starts to divide into two chromatids. So we now know that it is because of the S phase that the single DNA was replicated into two, and hence we got like this kind of structure. Okay, now this is at the center, centromere is there. Now once it is in the anaphase, what happens? They get divided into two. So one sister chromatid goes in one direction, the another sister chromatid goes into the another directions. Okay. Now each chromatid now called chromosomes and moves towards the opposite side, which is accomplished by two forces. So what are those forces? The poleward force, and the second is the polar ejection force. So this is what I was talking about. So you have got the, suppose let us say that this is your polar microtubule, onto which the you have got the motor proteins which is holding the chromosomes. They will be pulling, or they will be pushing the polar microtubules and going towards the centrosome. Now in the late anaphase chromosome, they reach the opposite pole. So this is divided into two. That is, the early anaphase is called the anaphase A, and the later one is called the anaphase B. Now what does it mean? It means in the early anaphase A, the chromosomes are being pulled apart, and in the anaphase B, that the cell is going to get divided into two, as it means that it is going to reach to the telophase phase of the mitosis. So now each centromere represents the centrosome here. centrosome gets converted into chromosome gets converted to chromatin fiber, and as I mentioned, so once these chromosomes are getting over here, the nuclear membrane will start getting assembled. Now, how exactly this is happening is, so we are having the vesicles of the nuclear membrane as mentioned before, okay, and now we have got the chromosomes. It is the single cystochromatic. So these nucleus or nuclear membrane vesicles, they go and bind to chromosomes like this. Okay. Now later what is happening? Then all these will be coming together. The vesicles will come together, taking chromosomes inside. So finally, what we are getting is actually chromosomes are present and these vesicles will merge together to form the nuclear membrane envelope. So this is how the nuclear membrane appears back. Okay. And obviously the nucleolus will also reappears in this stage. Now the cleavage furrow formation starts in this phase, which leads to cytokinesis. So cytokinesis finally means you are going to have the two daughter cells over here. Those are going to be identical. So in animal, all cleavage furrow formed by the actin and myosin 2. So what is myosin 2? It is your actually the motor proteins of microfilaments and these microfilaments are made up of actin. Okay, so they are made up of actin. So they are the motor proteins which drive the cytokinesis process. Now in the plant cell, cell plate is informed in the middle of the cell. Now this is made up of the vesicle derived from the Golgi apparatus. Now this is also very important. Okay. So in the case of animal, the cleavage furrow is formed by the actin and myosin 2 proteins, which are the motor proteins. Okay. In the case of plant, it is formed in the middle of the cell. Now these are formed by the vesicle formation, which are derived from the Golgi apparatus. So this is very important to remember. Now, mitosis is important for the maintenance of the chromosome set. Yes, it also occurs during the growth and development. Of course, when an organism is growing, what does it mean? It means that the cell is being divided repeatedly. Okay. Now, a single cell zygote is converted into multicellular body through mitosis. So हमारे बॉडी में भी हम इतना स्किन रिपेयर होता रहता है. It's all because of the mitosis. So in your hand also, every time you shed these cells, right, the dead cells are being removed and replaced by the new ones. So all this is happening and it is all possible just because of the mitosis. Okay. So फॉर एग्जाम्पल लाइक आप गर्म चाय पी रहे हैं एंड अचानक से जीभ जल गई है. ओके, सो फॉर सम टाइम यू डोंट फील एनी सेंसेशन, यू फील दैट द टेस्ट हैज बीन गॉन, बट अगले दिन क्या होता है, आपको वो टेस्ट वापस मिल जाता है, राइट? इट्स जस्ट बिकॉज इवन योर टंग इज एक्चुअली शेडिंग ऑफ द डेड स्किन ऑफ द टंग and it is being replaced by the new cells. So all these mechanism, it's possible because of the mitosis, and this mitosis is being happening continuously in our body. Okay. So tissue regeneration is also achieved by the mitosis. Asexual reproduction, like budding of the hydra in noble animal, is a type of mitosis only. Vegetative propagation in plant is also mitosis. It's just that whenever we talk about these, we don't say mitosis. We have named or we have been calling them with the different terminology. Okay. Now, if any error occurs during the mitosis, it can create aneuploid cells. That is when they have less amount of chromosome. That is, it is less than 2n. It is not diploid. Okay. Or they have too few or too many of more chromosomes. Okay. So finally what will happen is either the aneuploid cell will die or it will result in the formation of the cancer. So whenever you talk about cell cycle or cell signaling, if anything goes wrong or any protein is not functioning, just understand that everything leads to then the formation of the cancer. Okay. Yeah, so that's all for the today class. So today we talked about the different stages of the mitosis, जिसमें हमने पढ़ा प्रोफेज, मेटाफेज, एनाफेज एंड टीलोफेज. ओके, देन प्रोफेज में भी आपका अर्ली प्रोफेज होता है, लेट प्रोफेज होता है, Late prophase means where you are, the nuclear membrane is getting fragmented and finally you're entering the metaphase. Metaphase में क्या होता है, आपके क्रोमोसोम एक सिंगल प्लेन में आ जाते हैं. It's actually imaginary. There's no certain line present over there. Okay. Then after that, what happens? The chromosomes are being pulled apart. So then it is entering the anaphase. Anaphase में क्या होता है, आपके chromosomes separate हो जाते हैं, nuclear membrane वापस आने लग जाते हैं, and finally telophase में you get a two separate identical daughter cells. Okay, so that's all about the mitosis and about the mitosis regulations and the cycle CDK1 cycling the activity. This and all we have discussed in the previous lectures. Okay, so thank you for listening to this. I hope you like the lecture. Please keep watching, understand about the cell cycle. If any queries or doubts are there, please put into the comment box, or you can mail us at the excelsioracademy1228@gmail.com. You can also find and ask a query in the Telegram channel also. Thank you.